Inside scorpion venom, a future RX for arthritis? Tiny scorpion-derived proteins deliver arthritis drugs to joints in rat study. Millions of Americans live with the pain and physical limitations of arthritis and other inflammatory diseases. Steroids and other drugs can offer relief, but they can also come with a host of serious side effects. A new study suggests that a scorpion-derived mini-protein could one day help tame the sting of that toxic trade-off. Scientists at Fred Hutchinson Cancer Research Center identified a tiny protein in scorpion venom that rapidly accumulates in joint cartilage. Then they linked these mini-proteins with steroids to reverse inflammation in rats with arthritis. The researchers found that the drugs concentrated in the joints, potentially avoiding the body-wide toxicities and infection risks caused by non-target steroid treatment. The approach is several years away from human patients, said the project's senior scientist Dr. Jim Olson, who discovers and develops new drugs using blueprints from nature. But as a proof of concept, it is promising. For people with multi-joint arthritis, the side effects of controlling the disease can be as bad or worse than the disease itself, Olson said. Steroids like to go everywhere in the body except where they're needed most. This is a strategy to improve arthritis relief with minimal systemic side effects. Scorpion venom may cause severe medical complications and untimely death if injected into the human body. Neurotoxins are the main components of scorpion venom that are known to be responsible for the pathological manifestations of envenoming. Besides neurotoxins, a wide range of other bioactive molecules can be found in scorpion venoms. Advances in separation, characterization, and biotechnological approaches have enabled not only the development of more effective treatments against scorpion envenomings, but have also led to the discovery of several scorpion venom peptides with interesting therapeutic properties. Thus, scorpion venom may not only be a medical threat to human health, but could prove to be a valuable source of bioactive molecules that may serve as leads for the development of new therapies against current and emerging diseases. This review presents both the detrimental and beneficial properties of scorpion venom toxins and discusses the newest advances within the development of novel therapies against scorpion envenoming and the therapeutic perspectives for scorpion toxins in drug discovery. Keywords: scorpion venom, potassium channel toxins, calcins, scorpionism, fungicide, parasiticide, bradykinin potentiating peptide, analgesics, antivenom. According to national public health data, about 1.5 million scorpion envenomings, resulting in 2,000 to 3,000 deaths, are recorded annually worldwide. While large regions in the Northern Hemisphere, such as the United States, Canada, Europe, and Russia, as well as Australia in the Southern Hemisphere, are not associated with severe scorpionism, more than 2 billion people living in Northern Saharan Africa, African Sahel, South Africa, Near and Middle East, Southern India, Eastern Andes, Mexico, and South America are at risk of being stung by scorpions. Climate change, together with urban expansion and poor city sanitation management in many of these areas, have increased the likelihood of encountering scorpions. For instance, in Brazil alone, malign incidences with scorpions have nearly doubled from 64,000 to 124,000 annual envenoming cases since 2012. To date, over 2,000 scorpion species have been described. The vast majority of the scorpion species that are dangerous to humans belong to the Buthidae family, 5, but some species in the families of Scorpionidae and Hemiskirpidae have also been classified as harmful. Geographical distributions of most of these medically significant species correlate with the local prevalence of scorpionism. Hence, the density of dangerous species is especially high in northern Africa, Iran, Saudi Arabia, Brazil, Mexico, and Venezuela. Local pain is often the first symptom of scorpion envenoming, which may set in only minutes after a sting has occurred. Depending on the scorpion species, the symptoms can progress to severe complications over the course of a few hours. Inducing a massive release of neurotransmitters, scorpion venom neurotoxins usually cause sweating, nausea, vomiting, hypersalivation, restlessness, and, in more severe cases, arrhythmia, unconsciousness, and heart failure, which may lead to death. However, in spite of the hazardous and life-threatening effects of scorpion envenoming, therapeutic properties of scorpion body parts and venoms in ancient medicine have been utilized by humans for thousands of years. Nowadays, The potential therapeutic value of different scorpion venom compounds is being increasingly investigated, as these compounds may represent promising leads for the development of new pharmaceuticals. In this review, we survey the field of scorpion venom research from different angles, focusing both on the detrimental and beneficial properties of scorpion venom toxins. First, scorpion venom compounds, together with clinical manifestations and symptoms of different levels of scorpionism, are introduced. Then, currently available treatments and research into new alternatives, i.e., next-generation antivenoms, are discussed. Finally, the latest reported results from the scientific literature focusing on the widespread potential applications of scorpion venom compounds are presented. Scorpion venom compounds. Scorpions use their venom to defend against predators and to capture prey. The composition of scorpion venom is highly complex and heterogeneous. Up until now, small scorpion venom peptides are the most studied compounds, mainly due to their diversity and broad pharmacological properties. Accordingly to their structure, these small peptides are classified into three large superfamilies: peptides containing cysteine-stabilized, CS, alpha/beta motifs, calcins, and non-disulfide bridged peptides, NDBPs. However, enzymes, larger proteins, mixtures of inorganic salts, free amino acids, nucleotides, amines, and lipids are also found in scorpion venom. These peptides consist of an alpha helix joined to a double or triple-stranded beta sheet via a disulfide bridge. These molecules present two completely conserved disulfide bonds in the CI, CJ, and CI+4, CJ+2 positions, although some of them also exhibit an extra link connecting the two endings of the peptide chain. All scorpion peptides containing CS alpha/beta motifs act in a similar way. Their interaction with ion channels result in blocking or modulation of the normal mode of action of these channels. Members of this superfamily can be subdivided into long or short scorpion toxin families corresponding to their respective structures. Peptides from the long scorpion toxin superfamily are 55 to 76 residue-long molecules with generally 4 disulfide bridges due to their mode of action. They can also be called sodium channel toxins, NaTXS, as their main targets are sodium ion channels. This family can be further divided into two groups, alpha and beta NaTXS, depending on their specific interaction with the voltage-gated Na+ channels. Alpha NaTXS block site 3 of sodium ion channels and therefore inhibit the inactivation of the channels and prolong their action potential. In contrast, beta NaTXS interact with site 4 of Na+ channels and shift the activation voltage of the channels to a more negative potential, which results in channel inactivation, opposite to the effects of site 3 toxins. 14. It is noteworthy that not all NaTXS can be assigned to these two groups. For example, CN12 from Centauroides noxius and TS2 from Titius cerulatus venom structurally resemble a beta NaTX but exhibit an alpha NaTX effect. In addition, IT4, a toxin from Androctonus australis hector, displays both alpha and beta NaTX effects. The short scorpion toxin family is composed of peptides of 23 to 64 residues in length with 3 or 4 disulfide bridges. These peptides, also known as potassium channel toxins, KTXS, mainly act as potassium ion channel blockers. Considering their sequences and cysteine pairs, KTXS can be divided into alpha, beta, gamma, kappa, delta, lambda, and epsilon KTX groups, 18, 19. The alpha KTX group, which is considered to be the largest subgroup of the short scorpion toxin family, contains 23 to 42 residue-long peptides with 3 or 4 disulfide bridges. The beta KTX group comprises longer-chain peptides of 50 to 57 residues in length. The gamma KTX group can be found in the genera Centauroides, Mesobuthus, and Buthus, and it mainly blocks the human ether-ago-gorylated gene, HERG, channels. Instead of having a CS alpha/beta structure, similar to the alpha, beta, and gamma KTX subfamilies, toxins of the kappa KTX group are composed of 2 parallel short alpha helices connected by a beta turn that is stabilized by 2 disulfide bridges, yet their interaction with potassium ion channels is similar to that of the alpha KTX group. Although the delta, lambda, and epsilon KTX groups do not contain a CS alpha/beta motif, they are mentioned in the continuation of other KTXS. The delta KTX group contains a Kunitz-type structural fold with a double-stranded antiparallel beta sheet flanked by an alpha helix in both the C-terminal and N-terminal segments. Since the Kunitz-type structural folds are the active domains of proteins that inhibit the function of serine proteases, delta KTXS exert both protease and potassium channel inhibiting properties. The lambda KTX group, similar to calcins, contain an inhibitor cysteine knot (ICK) motif that contains a triple-stranded antiparallel beta sheet stabilized by 3 cysteine linkages. The epsilon KTX group has recently been defined and, so far, has just 2 members, TS11 and TS12 from T. serratus Venom. TS11 shows less than 50% identity with KEXS from other subfamilies. TS11, similar to lambda, KEXS, contains an ICK motif. However, lambda KTXS possess only 3 disulfide bridges, while TS11 has 4 disulfide bridges assembled in a unique pattern. This small, but growing, family of scorpion toxins consists of calcium channel-modulating peptides such as impercalcin, impertoxin, morocalcin, hemicalcin, hadrocalcin, opicalcin, urocalcin, and vigocalcin. Sharing high sequence similarity, greater than 78% identity, calcins include an ICK motif stabilized by 3 disulfide bridges. Calcins mainly act as agonists of ryanodine receptors, RERS, which are intracellular ligand-activated calcium channels that are found in endoplasmic/sarcoplasmic reticulum membranes. RERS play an essential role during excitation, contraction coupling in cardiac and skeletal muscles by releasing Ca2+ from intracellular reservoirs. In general, calcins induce long-lasting subconductance states on the RER channels, which lead to an increase in the intracellular Ca2+ level and, subsequently, contractile paralysis. Calcins also present the ability to pass through cell membranes without causing their lysis. 31. It has been hypothesized that the clustering of positively charged, basic residues on one side of the calcins gives them a dipole moment that possibly interacts with negatively charged membrane lipid rafts, such as gangliosides. Once these toxins interact with the outer membrane, interaction between the hydrophobic regions of the toxin and the inner membrane is favored, and the toxin is transiently translocated. Further electrostatic interactions with negatively charged molecules from the cytoplasm trigger the entrance of calcins into the cell without disrupting its membrane. 32. This feature makes the calcins excellent candidates for intracellular drug delivery, since they can enter cells without disrupting them, even when large. Membrane-impermeable molecules are conjugated to them. A calcium channel modulator, distinct from the toxins that act on RERS, was recently identified through transcriptome analysis of T. serratus and designated as a cell-penetrating peptide, CPP-TS. The synthetic CPP-TS is the first described scorpion toxin that activates Ca2+ signaling through the nuclear inositol 1,4,5 trisphosphate receptors. This toxin, together with the calcium channel toxin like BMCA1 from Mesobuthus martensii, forms a new subfamily of calcium toxins and shows promising anti-cancer effects. It is noteworthy that calcins are not the only cell-penetrating scorpion toxins. The transient receptor potential cation channel subfamily V, member 1, TRPV1, is a chemosensory ion channel, which is also known as the wasabi receptor. Generally, TRPV1 is activated through a unique mechanism involving the covalent modification of specific cysteine residues located within the channel's cytoplasmic N-terminus. Recently, it has been reported that wasabi receptor toxin, WATX, from Urodicus manicatus venom, is capable of activating this receptor. This means that WATX can cross the plasma membrane and bind to the same allosteric nexus that is covalently modified by other agonists. NDBPs are small, 13 to 56 amino acid long peptides with a very heterogeneous composition. Compared to scorpion peptides with disulfide bridges, NDBPs do not present a conserved or predictable structure-function relationship. Most of these peptides are cationic molecules that display notable structural flexibility. In aqueous solutions, these peptides exhibit a random coil conformation. However, under membrane-mimicking environments, such as 50% to 60% of aqueous trifluoroethanol, they readily adopt an amphipathic alpha helical structure. This characteristic enables them to interact with a broad spectrum of biological targets. However, they do not have any known specific molecular targets. Few enzymes have been found in scorpion venoms, in part because up until recently, interest has been focused on small proteins and peptides. However, during the past years, hyaluronidases, phospholipases, and metalloproteases, among other enzymes, have been detected in venoms of different scorpion species. Different hyaluronidases have been identified in different families of scorpions, including Buthidae, Bothriuridae, and Urodacidae. It is known that these enzymes potentiate the toxicity of venom by disrupting the integrity of the extracellular matrix and connective tissues surrounding blood vessels at sting point, and they thereby ease the systemic diffusion of other relevant scorpion toxins. It has recently been demonstrated that hyaluronidases also play an essential role in venom distribution from the bloodstream to the target organs. The same study also indicated that the neutralization of hyaluronidases could be considered as a first-aid strategy in scorpion envenoming treatment. Phospholipases are known to be potent hemolytic agents, as they disrupt cell membranes by hydrolyzing phospholipids. They can also cause tissue necrosis and hemorrhages. Phospholipase activity has been detected in several scorpion species, including Opisthocanthus cyaporum and Heterometrus laeoticus. Generally, venoms from Titius species exhibit significant proteolytic activity, and the first scorpion metalloprotease was discovered in the venom of T. Cerulatus metalloproteases and serine proteases have also been detected in T. discrepans venom and, apart from Titius species, in the venom of Hemiscorpius leptris, it is believed that proteases play a key role in activating toxin precursors through post-translational modifications. In addition, these enzymes inhibit platelet aggregation, modulate cytokine production, and activate the complement system altogether. These effects facilitate the diffusion of scorpion venom toxins via the degradation of matrix proteins. Only a very limited number of studies on non-peptidic scorpion venom compounds have been reported. However, it has been shown that the venoms of some scorpions, including M. tamulus, contain serotonin, which is a monoamine that may cause vomiting and considerable local pain in scorpion envenomings. The metal and salt composition of some scorpion species have also been evaluated by Al-Azmari et al. In their study, they found copper, zinc, calcium, magnesium, iron, lead, manganese, arsenic, and nickel ions in the venom of A. bicolor, A. crassicata, and Liurus quinquestriatus. They suggested that these components are associated with enzyme activity, as they probably act as enzyme cofactors. In addition, 2,1,4-benzoquinone compounds, with antimicrobial activity, have recently been isolated from the venom of Diplocentris mellici, a scorpion species endemic to Mexico.